אחרי צהריים טובים. I am Raphael Levine. It is my pleasure, and also I am somewhat sad to announce the last session. Why am I sad? I learned so much this week that I'm sad that it's coming to an end. Maybe the president will allow us a repeat performance in another 20 years. I hope so. And we have a very busy afternoon, so I have a pleasure to introduce Bill Schopf, Earth's oldest fossils. What's the experimental evidence?
Thank you, Rafi. On behalf of us all, I'd like to thank, of course, Rafi and El Presidente, and the Academy of Sciences and Humanities of Israel. This has been a splendid get-together. And yes, we've learned a very great deal. And it has been a wonderful experience. So I thank you very much. I also want to thank George Whitesides, because George introduced a concept early in these proceedings, suggesting to us that we should have a look at and in fact read the book by Thomas Kuhn, The Structure of Scientific Revolutions, 1962, University of Chicago Press. Now, it turns out that has influenced me a great deal as well as it has George, and I happen to have met Tom Kuhn. And in fact, he has urged me to write a book and to essentially, some of which I'm going to talk about today. His book was largely in part on the Copernican revolution and the Darwinian revolution. And the revolution I'm going to tell you about, paradigm shift, pales into insignificance in comparison with those, of course, but it is the revelation of the earliest 85% of the history of life, so it does have some significance, both in the sense as well that it has spawned two new fields of science, one called Precambrian paleobiology, and the other called astrobiology. And I can assure you that everything that is going on right now for the planning for the Mars 2020 mission is based on the work of this field. That's why we're looking for microscopic organisms to discover the past history of life on Mars and on other planets. And so it is a significant change in human knowledge. But it's not like the Copernican revolution, you know. I mean, let's be real about this. Okay, now I got a thing here. I push that, and it says boom. And it doesn't say boom, but it does so. The story starts, of course, with Charles Darwin, 1859. Now, Darwin writes The Origin of Species, finished in 1844. Published in 1859, largely because of a letter he received from a biologist doing, Alfred Russel Wallace, doing biology and geobiology, or doing biology and geographic distribution of organisms in Malaysia. That had the same idea that he did, and thanks to Hooker and Charles Lyell, they arranged a joint presentation at the Linnean Society of London in July 1st of 1858. It did not cause much of a problem, or nobody really cared until Darwin's book came out. This was from his manuscript, and this is the first page of the title page from the first edition. Okay, so Darwin comes along, and what a lot of people don't realize is that the theory of evolution is based largely on the fossil record. It was known because of the cutting of canals through England and parts of Wales and parts of Eastern Europe and so forth, but largely in England because of the Industrial Revolution. The goods would come in from the colonies at Plymouth in the south, and then they'd have to be transported north to Manchester to be made into goods to sell back to the people in the colonies, and that's the way the Industrial Revolution worked. Okay, but in that process, William Smith discovered a whole lot of fossils and codified all these things. So by 1849, which is before, 10 years before Origin of Species, you see things like this lithograph, which outlines the organisms of the Paleozoic and the Mesozoic and the Cenozoic. Now, based on the fossil record, it was a good theory, but the fossil record also caused various problems. And as Darwin says, he says, if the theory of evolution be true, it's indisputable that before the lowest Cambrian stratum was deposited, the world swarmed with living creatures. Yet, while we do not find rich fossiliferous deposits belonging to these assumed earliest periods, I can give no satisfactory answer. The case at present must remain inexplicable and may be truly urged as a valid argument against the views here entertained. And it was this way for the next 100 years. And my professor in college said that this was the greatest unsolved problem in natural sciences, which got me interested because I thought he was wrong. He was right. All right. Sometimes the professors are, they get lucky. All right. Well, I'm one of those guys too, All right. And here's the nature of the problem. The oldest fossils known were trilobites, which are arthropod and lobster-like organisms, much too complicated, many-celled eukaryotes that could have nothing whatever to do with life's earliest stages. The problem then was what happened before that, and it was unknown, and for a full century up till 1965 was in fact regarded as unknowable by the world's scientific community. Well, we now know that's the age of microscopic life. Now, for many of you, you're not used to thinking about geological time, and I don't blame you. A million years seems like a really long time, but then you talk 10 million years, and you talk 100 million years, and you talk 1,000 million years, 1 billion years, and I'm going back 3.5 billion years, and how can anybody understand any of that stuff? So what I've done is taken Earth history and condensed it down to one 24-hour day. And if that's the case, the formation of the Earth happened last night at midnight. And it would follow from that the origin of life occurred this morning at around 4 o'clock while you were asleep. The oldest fossils were entombed at around 5:30 this morning, followed by a long period of microbial evolution, asexual things, so it's very slow. You get up to about 9 o'clock, and then you get the trilobites, and then at 10 o'clock, land plants, at 11 o'clock, dinosaurs, at 11:40, mammals come in, and humans come in 17 seconds before midnight. All right, well, what Darwin knew was from 9 o'clock tonight until midnight tonight. What we now know goes back to 5:30 in the morning. And that is the paradigm shift that I'm going to address. As you'll see, the history of this field has been circuitous, but you know your own science, it always is. At the same time, it's instructive to note that cultural biases play a role in our science, or at least in this science, and not only religion, because after all, this is about evolution, but also international politics, which most of us don't think much about. Both of those cultural biases make a difference here. And then there's a third thing about which each of us in this room should guard against, and it's what I call science by authoritative assertion. Because if you're a biggie big, folks believe you, and that means you have a responsibility to be especially careful about what you say, because they will pick it up because you're supposed to know, and then it turns out that, by golly, you were mistaken. And so in this case, I'll tell you the story, it set back the field by 30 years. All right, well, it starts off with Charles Darwin, who states the problem in 1859. Immediately thereafter comes John William Dawson. Now, this is 1865. He'd been given he was a real luminary. He was the president of a whole bunch of societies and was the principal of McGill University. In 1855. He was not the first principal of McGill, but he is the one who is given credit for establishing McGill University in Canada to its state of prominence, which it still has in Canadian education. So he was given samples by Logan, who was the director of the Geological Survey of Canada, and he started studying them. Now Dawson was a Calvinist Christian his parents came from Ireland. And he was an anti-evolutionist and a staunch diluvialist, and he was a staunch creationist. All right, so he's given these samples that came from down here in the Ottawa River. He starts studying these things, and it is this sort of a structure. This is from his 1875 book. In 1865, he names this Eozoan canadense, the dawn animal of Canada. Now, he writes a book in 1875. Here's a specimen, here's a sample of that specimen that I photographed at the U.S. National Museum, because I wonder really what he was up to, and there it is. He writes a book in 1875 called The Dawn of Life, in which he explicates Eozoan canadense in some detail. And he indicates that he thinks it is a foraminiferal protozoan, which is rather striking because this thing's the size of a volleyball. But in any case, he goes on, magnified and restored section of a portion of it with the brown matter showing the animal matter in the chambers and the canals and the tubuli and the pseudopodia and so forth and so on. It's really quite an interesting book. But the problem is that his sample is nearly 600 times larger than the largest protozoan fossil known to man or beast. Yeah, but I didn't slow him down. He decided that's what it was. So he publishes this thing in 1865. He thought, and you ask why, if he's a creationist, well, he decided this was proof of special creation, biblical special creation, and therefore disproof of evolution. And he writes, he says, there is no link whatever. This thing is 1 billion years old, and the oldest fossils are 500 million. And he says there's no link whatever to connect Eozoan with these younger fossils. All stand before us as distinct creations. And then he goes on, Eozoan thus bears damaging negative testimony against evolution, which is incapable of proof and contrary to fact. In fact, evolutionists are mere dreamers, having no scientific basis for their dogmas. So there's culture modifying the view or dictating the view of a scientist. Same thing happened to Baron Georges Cuvier when he first discovered extinctions. Science is replete with such examples. Well, in 1865, he describes this thing. In 1866, King and Rowley, who are two Irish mineralogists, look at the paper and say, no, 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 this is purely mineralic, it's non-biological. Not to be dissuaded, he sends samples to Carl Moebius in Germany, who is arguably the world's expert on foraminifera. And Moebius says, well, they're certainly not biological. And then in 1894, Gregory and Johnson Lavis from what was then the British Museum of Natural History, now the Natural History Museum in London, find this Eozoan on the flanks of Mount Vesuvius outside Pompeii, and indicate that it's a serpentinized limestone, which is a metamorphic change from a normal limestone. It's the sort of thing, if you go to the United States, go to a bank building, look at their floor, and you'll see the serpentinized limestone all over the place. It's a real pretty green and white rock. It has nothing to do with life. And so he has the dubious distinction of describing the first pseudo-fossil, but he believed this up until his death of 1899, and he only set the field back by 40 years. All right, so we're through that, and then comes along the real hero who has the right idea, and that's Charles Doolittle Walcott. Now, Walcott was quite an individual. He was the president of the American Philosophical Society. He was director of the U.S. Geological Survey. He was president and secretary of the Smithsonian Institution, which meant he was the boss. He was a confidant of two American presidents. He was a mover and a shaker. He only had 10 years of schooling at Utica High School in northern New York State. At the age of 26, he was hired by James Hall, who was the New York State geologist, and at that time, in 1875 and 1876, New York was a real important place in the United States, New York State, and this guy was the chief geologist in that. And so this is before the establishment of the U.S. Geological Survey. This was the guy who was the sort of the major geologist in the whole country at that time. So he only worked for him for 3 years, but what was interesting was that Hall, in 1870, had gone over to Saratoga Springs in New York and had found, they'd been reported in 1825, but he went over and looked at them and he says, oh, I think these things are biological. They're concentric structures. He named them cryptozoan, and here they are. They're in the Cambrian, 525 million years in age. He interpreted these things as algal reefs, but nobody else in the world would believe him except Walcott. Walcott went over there in 1870, and he agreed that these were biological. All right, so comes along, what are they? They are what we now call stromatolites. They are microbial mat communities. You can see the laminae here. This is a modern mat community, and at the top, typically these days, it used to be photosynthetic bacteria. Now they are cyanobacteria, oxygen producers with the photosynthetic anaerobes just beneath them. In any case, we know what they are now, but that was because the modern analogs were first described in 1961. Okay, and here you can see the similarity between the modern stromatolite microbial mat on the left and a fossil stromatolites 1.3 billion years from the Belt Supergroup in Montana at 1.3 billion years. All right, you see the similarity of them. And in 1879, Walcott went on the first expedition down the Grand Canyon, the Colorado River, the Grand Canyon of Arizona. It was a harrowing trip. They went down on log rafts. You read the history of this thing, it is just mind-boggling. In any case, he went down there and went down the Grand Canyon, and there in 1883, on his second trip down, he did this repeatedly, He discovered stromatolite ridge and these stromatolites, the same structures he'd seen at Saratoga Springs. Okay, so he knew what they were. He'd already decided they were biological. Then in 1899, on another trip, he discovered the first Precambrian microscopic fossil. He misinterpreted it. He thought it was the crushed shell of an invertebrate animal because that's what the going dogma said. It's not. It's a planktonic unicell. It's called Chuaria, and he named it, and we have lots and lots of examples nowadays, but then this was the first thing, okay? He then went up the spine of the Rocky Mountains from the Grand Canyon up to the Lewis Range, named after Meriwether Lewis of the Lewis and Clark expedition of the early 1800s. And there he found another set of stromatolites. Okay, so he's got some good data, you think. He also thought that he'd found fossil bacteria. That was a good idea. He was mistaken. I've seen the specimens at the U.S. National Museum, and they are pseudofossils. They're mineralic blobs. But anyway, he had the right idea. He was asking the right questions. And from there, he moves up into British Columbia to Burgess Pass, which he names, and he names the Burgess Shale. And in that Burgess Shale, here's the Burgess Quarry today, and there is C.D. Walcott on the left. And this is the thing that you read about in the textbooks, the explosion of life at the Cambrian, where all of these major phyla of invertebrates and even hemichordates are known from. It's a wonderful deposit. And it's particularly marked because, striking because it has soft-bodied parts of these organisms preserved. We now have even better examples from northeastern China in Liaoning province, and also in southern China. So there's been a lot of progress there, but this was the first big discovery. Okay, so here it is, the Cambrian explosion of life, and it fit what was known before. Namely, you knew that there were trilobites there. Now you see all these new things coming in, and it fit previous dogma. Here are a bunch of 4 examples of the sorts of things that Walcott named. Carnivorous worms and velvet worms and slug-like animals and trilobites and things like that. And you know this is an important discovery because, after all, it more recently has made the cover of Time, the weekly news magazine. So you don't get stuff in the cover of Time, the weekly news magazine, unless it makes some sense, you know. It stood the test of time. Not only that, my friends, here in 2009, 100 years after his discovery, the Big Rock Brewery of Alberta, Canada, comes out with the Burgess Shale Centennial Celebration Ale. Now, I dare say that there are few of us in this room who are going to have a beer named in our honor 100 years after our first big discoveries. I'm probably mistaken. There are probably going to be a whole bunch of beers named after you guys. Anyway, there he is. He did everything. Stromatolites, algal unicells, stromatolites again, the Cambrian explosion of life, fossil bacteria, which was a mistake, and the shale ale and all that kind of thing. I mean, golly gosh, this guy was really, really good. Yeah, well, Walcott made the first major discoveries, and then progress abruptly stopped. Now that you ask yourself, you know, here we got this paradigm shift that's on the cusp. What happened? Well, comes along Sir Albert Charles Seward. Now, Seward, you know that Walcott's a big guy, you know. He's a biggie big. So it takes another biggie big to take him down. Yeah, well, Seward fit the bill. Seward was the vice chancellor of Cambridge University, which meant he ran Cambridge University. He was the CEO, all right? Yeah, but more than that, and he was, you know, knighted and all that, and that makes him very important, very well known, very much respected. But he was the world's leading paleobotanist, and these reports of Walcott's of supposed bacteria and these algal reefs, stromatolites, fell within the purview of paleobotany. Now, Seward was a prolific writer. 1931, he comes along with his book called Plant Life Through the Ages, and this is still when the sun never set over the British Empire, you know, and so everybody in the world read this thing. Very, very influential, from the world's leading paleobotanist, and he says, Walcott's interpretation of cryptozoan is, I venture to think, not justified by the facts. It's clearly impossible to maintain that such bodies are attributable to algal activity. We can hardly expect to find Precambrian rocks any actual proof of the existence of bacteria. If I had more time, I'd tell you why he should have known better, because of works published in 1917 and 1921. The Kidston and Lang work on the what the heck? I mean, there are a bunch of bacteria, cyanobacteria, in those publications, and I know he knew about the books because he writes about them, but he didn't read the last of the 5 volumes, and that's where they're hidden, in with fossil fungi as well, and he should have known better, but he didn't. Anyhow, so he says that, but everybody listened to him. And then he comes up with these poems. The guy was very, very creative. I never met him, but I know his students. I've met some of his former students. And he says, talking about Precambrian fossils, he says, creatures borrowed and again conveyed from book to book, the shadows of a shade. Which I thought was pretty cool, you know. Anyway, there it is. He's a little doggerel to put down the other guy. You rarely see that anymore, I hope. But in any case, Walcott's Cambrian Explosion was accepted, but these Precambrian fossils were all rejected because of Seward. Well, then nothing happened until progress ceases entirely, until you get up to the 1950s, about the time that the State of Israel is getting established. Okay? And that was work of Boris Vasilyevich Timofeyev. Now, A.C. Seward, science by authoritative assertion. You believe the big guy. Well, now we come back to a cultural thing that held us back, held the field back. Timofeyev was in Leningrad. He was a Soviet. Now, what he'd found, he misinterpreted these things too. These are 1 billion years old, 1.3 billion years from Siberia. He thought they were the spores of land plants, but land plants do not come in until 420 million years ago. So that was way off base. Nevertheless, they're real good fossils, and they've done some really good work. But the problem was that there were Cold War politics going on between East and West. And it turns out he was a commie. No, he really wasn't. I spent 2 months in his lab. He wasn't. He'd been put in the gulag. The only reason they let him out was because his way of getting fossils out of rocks could be useful to the petroleum industry in the Soviet Union. But then he told me he'd been in the gulag, but we're sitting there in his office, and he goes like this. I had no idea what he meant. And he says, he took me to a park across the river where nobody could listen in. He was afraid that he knew very well that he was getting taped, that he'd get in trouble. And so then he told me about the whole business. And they wouldn't let him travel. He couldn't get out of the country and so forth and so on. But in any case, in the West, A, he was writing in Russian and nobody could read Russian. B, My professor refused to reference any of his papers, however relevant, because he was a godless commie. I'm sorry, that's a direct quote. And so, look, I mean, this is something we don't think much about. But these cultural biases can hold us back. And I personally don't think science knows boundaries, shouldn't know boundaries, shouldn't matter, in my opinion, what tribe a person's in or what stage of development their country happens to be or what the color of their skin is or whether they're male or female or what. The fact is, facts gotta matter, folks. Facts gotta matter. It doesn't matter who, not the messenger, it's the message that matters. Anyway, that's the way I look at it. Okay, on I go. Okay, so then we go up to 1965. Well, my hero, my professor, Elso Barghorn and Stanley Tyler. Tyler in 1952 had discovered fossils that turn out to be about 2 billion years in age. Professor Barghorn had had some personal difficulties, so he didn't do anything for 10 years. I showed up. Tyler died before I could meet him. Barghorn went out to University of Wisconsin, brought everything back to Harvard where I was doing graduate work, handed it over to me and said, sort it out. So I had to sort out all this stuff, and it was kind of arduous, but it wasn't too bad because I had done an honors thesis as an undergraduate on the same deposit and was quite familiar with what was going on and all that. So I helped Barghorn prepare this paper that comes out in 1965, and by that time I'd discovered fossils in another rock of another age in the Precambrian of Australia, and that came out 6 months later, and it was those two papers that set the field on its current path. The Barghorn-Tyler paper is shown here. These are fossils 1,900 million years in age. The problem with them is that they were 4 times older than any fossils known on Earth. And so even though the data were very good, they were met with incredible skepticism because ever since the time of Darwin, teacher to student, teacher to student, teacher to student, teacher to student, this is unknown and unknowable. So there must have been some awful mistake. But you folks don't know that by that time I already had another deposit coming up, this one here, for 850 million years in age from Australia. Barghorn knew that. He knew he'd be in the clear as soon as we got the second paper published because it would show this was no fluke. And that came out, and those 2 papers within 6 months in Science magazine set the field on its current course. Well, it turns out, I just was lucky. I happened to be there at the time. It turns out that that was 1965 stuff, and here is the history of this field in a nutshell. You see that going up the ordinate, and it does not matter what you plot there, whether it's workers, time spent, discoveries, fossils found, species named, publications, or whatever. That is the history of this field. And we've been articulating that change of paradigm, that's Kuhnian talk, Tom Kuhn stuff, ever since those breakthrough discoveries. Well, now there are thousands of fossils known, hundreds of workers around the world, and from Australia, Brazil, Canada, China, England, South Africa, Russia, all those stars and lots of other ones. Those are the ones that came to mind as I put that slide together. So I can tell you we're making progress. Now I'm going to flash forward to the present or the near present. 1993, doing field work and stuff. We got some fossils. I got some fossils from rocks 3465, nearly 3,500 million years, 3.5 billion. From Western Australia. Now, I'm not going to have a lot of time to talk about the whole deal here, but let me point out that I measured the cells in 1,985 specimens, and based on their morphology, divided them into 11 different systematic taxa, that is, species, we call them. But the point is there are a whole lot of them, and the reason that that matters is that biological systems you don't get a monospecific assemblage living in nature. There are always other things around, and so there are a whole bunch of things there. And so, one, there are many specimens. Two, the fossils are relatively abundant. Here is a field of view that shows 4 of them. Well, it's not quite enough because so they look like fossils. Are they fossils? Well, first thing you got to do, you got to figure out, are they really made out of carbonaceous matter? How do you do that? Oh, well, okay, so we developed in my lab first application to fossils of confocal laser scanning microscopy. And what it does, it causes the polycyclic aromatic hydrocarbons in the organic matter, their cell walls, to fluoresce. And so here are a couple specimens, okay? Well, you know, there are some minerals that fluoresce too, so maybe that ain't the deal. So then we used, again, we were the first to use Raman spectroscopy. And Raman spectroscopy is quite good. It definitively identifies carbonaceous matter. And here you see 3 specimens in the Raman spectrum and all that. Okay, so they're made out of carbonaceous matter, but that's not enough. If these are microorganisms, they've got to be cylindrical. And they have to have cells in them. How do you do that? Okay, so we figured out how to do 3-dimensional Raman spectroscopy. And here's one example, and you can see the cell lumina, which are infilled by silica, going down through a given filament. And you can also see that indeed that it is a cylindrical garden hose-like arrangement, which is what it ought to be. Okay. So you got some good fossils there. You met all those 3, 4, 5 different cascade of evidence, but do they stand alone? No, they don't stand alone. As you can see, this is from some years ago, but you got stromatolites and microfossils and carbon isotopes and Raman spectroscopy and stuff from a bunch of other deposits, 3.2 to 3.5. So we got plenty of evidence. Okay, well, it seems like you're nailing it pretty well, but how'd they live? Well, you want their physiology. All right, so I had set up an international interdisciplinary group of 48 people from 8 countries who came to UCLA and worked in my labs 2 different times for 14-month periods, and I was real lucky to be able to get the funding and able to get these guys to come, and they were real good. And we did a whole lot of bulk analyses of carbonaceous matter. Now, thanks to Mark Tiemann, who's already introduced carbon isotopic measurements. I don't have to this group go over this in any great detail, but they're the 2 stable isotopes of carbon are carbon-12, carbon-13. There's a carbon dioxide capturing enzyme called ribulose bisphosphate carboxylase oxygenase, which has too many syllables for most of us to pronounce, so we call it Rubisco. But anyway, it turns out that the lighter isotope gets into organic matter. And the systematics of it, the carbon dioxide dissolves in the world's oceans, reacts with bicarbonate that is formed and with the calcium that comes off the land surface and precipitates calcium carbonate, which is the way we want to get rid of the CO2 that's building up from internal combustion engines. Unfortunately, we've already fouled up the system so much that it would take between 1,000 and 10,000 years for the geochemical reaction to take out all the stuff we put in during the Industrial Revolution. But that's another lecture. Okay, so we get that CO2 down into carbonates, and at the same time, Rubisco is capturing that CO2 and making organic matter. Those guys die, they get into sediments, so now you look at sedimentary organic matter called kerogen. You compare that with the carbonate carbon, and there is a 2.5% or 25 parts per thousand difference between those. Okay, that's a signal of photosynthesis. So we've done thousands of measurements and a whole lot more from petroleum companies from where there is petroleum. And here you see carbonate going back through time. You see organic matter going back through time. There's 25 parts per mil separation. It tells you photosynthesis was on this planet since 3.5 billion years ago. Yeah, but those are bulk samples. 1 kilogram of rock, you dissolve it down and you get the carbonaceous matter out so you can get the isotopes. That doesn't tell you what the individual organisms are because it's a mix of stuff from an ecosystem. That's not good enough. So then in the year 2000, I got lucky once again. I tell you folks, I'm probably the most fortunate person on the face of the earth. I really am. Kevin McKeegan in our department had rounded up the enormous amount of money that was required to get a secondary ion mass spectrometer. They were using them on zircons, which, you know, is important in terms of the early environment. And we finally got some time on it, and so that we could look at fossils. So we were the first guys to do that, too. Now it's throughout the world. For those who don't know about SIMS, there's one mass spectrometer produces a primary ion beam. It sputters off the surface of the sample that is off your fossil and produces secondary ions, and that's what you measure by their mass to charge ratio, and you can separate out these isotopes. Well, with the unit that we first worked on, I chose because we had lots of bulk analyses too. It is the work of Chris House, a PhD student who's now a professor at Penn State University. And here's an example of some of the fossils we looked at. We had 32 bulk analyses of that particular geologic unit. We did 15 microfossils, and you can see the two have the same sorts of ranges, and this being the first study that anybody done like this, it gave us confidence that we knew what we were doing. Okay, having done that particular thing, now you look at the analyses throughout the Precambrian. You see photosynthetic cyanobacteria and photosynthetic bacteria around -25 on that scale. Precambrian kerogens, that's the most of them, like 90–95% of them. They match, and that's just fine. But then in 1983, we discovered these. We first discovered them in 3 basins, but then there are 5 others, and it turns out that they're much more enriched than photosynthesis will do, enriched in the lighter isotope. What in the world were they from? Well, it turns out that archaeans produce, members of the archaea, methanogens, they produce methane, and that methane's very light, even lighter than they are, and it gets eaten up by things called methylotrophs that use it as their carbon source, and that's what we think it got into the sediments. So it's archaean methanogens giving rise to this carbon-12-rich methane, which is then metabolized by the methanotrophs and gets into the organic matter. And interestingly, it only occurs earlier than 2.2 billion years ago, and that's when oxygen rises. So we thought, okay, maybe these things are all, it's an anaerobic world, which of course it turns out to be by lots of data since that time. Okay, so we got these guys there. Now, you look at these 3.5 billion year old fossils, and you see that they are isotopically light as well. And so we looked at 11 specimens of 5 species that were defined on their morphology, and it turns out that 2 of those species had a delta C13 at -30, one at -35, and 2 more at -40, and those are much too light to be from normal photosynthesis, not oxygen-producing photosynthesis in any case. So you look at with their distribution, looks to us anyway that the ones around -30 are probably photosynthetic bacteria, and these other guys have got to be methane producers at -35 and methanotrophs at -40. Now, these are the first archaea that had ever been discovered in the geological record. We had the isotope record, but nobody found any fossils of them, and it's kind of annoying because that's called the Archean. And the Archean part of geological time, there were no archaea. Hey, we got archaea in the Archean. All my friends are, you know, they're real happy about that. I know it's kind of obtruse, but anyway, you know, we're easily satisfied. All right, so photosynthetic bacteria taking in hydrogen sulfide as their hydrogen source to hydrogenate CO2 to make sugar, essentially. And so they're around -30, and then you get these guys, the methanogens. They're producing. Their bodies are at -35. The methane they produce is around -50. It gets incorporated into methanotrophs at -40, and that's the story. And what's nice about it is that all three of those lineages are near the base of the rRNA tree of life. And that was work that we published in January of this year, those new isotope data. But last September, I published another paper from 3.4 billion-year-old rocks that show that there are sulfate reducers, sulfide oxidizers, and photosynthetic bacteria at 3.4 billion years. And that tells you that these lineages had already divided the archaea from the bacteria by 3.5 billion years. We've got quite a lot of diversity, and this is just the tip of the iceberg, because all this stuff is new. And we got 3 bacterial consortia, the first consortia that any paleontologist has ever seen in their lives. That's all in the last 3 years. So we're going fine. It's been 160 years since Darwin, 110 years since Walcott, 60 years since those breakthrough discoveries. And my bottom line is that primordial life evolved early, far, and fast. And with that, I thank you. I am asked, would I take some questions? I could say no, but of course I'll be glad to have some questions. What can I? Well, if you don't have any questions, let me. Yeah, I see one back there.
So, if you're going to look for life on Mars, what lessons are you going to apply from these early life on Earth?
Well, it turns out it's very nice of you to ask that, and I have some experience at that, as it turns out. In 1969, we got the samples back from the moon, and NASA had established a 6-person team called the Lunar Sample Preliminary Examination Team. While the astronauts were in quarantine, the 6 of us were on the other side of the biological barrier analyzing those things. So I know exactly what's going to happen when those things get back. The rocks will be returned. They're going to be cached by Mars 2020. The rock types that they have decided to collect are based on my work. So I've been told. I mean, about 4 or 5 years ago, I discovered microscopic bacteria preserved in gypsum, which is widespread on Mars, and that's the reason I looked at it. And it's something a paleontologist would never look at because it's a salt, it's easily washed away. But up in the Alps of northern Italy, there's the world's largest gypsum mine. It used to be the bottom of the Mediterranean. It's been uplifted with the Alps, so it's not getting washed away now. It's being used to make buildings and lots of other stuff, the casings for buildings. In any case, so I got a bunch of samples from there and then 8 other samples around the world, and discovered that, yes, you can get microscopic fossils preserved in gypsum. Nobody knew that. Nobody had asked the question. So they're going to be looking for that. They're going to be looking for cherts, which are silicon dioxide rocks just like that. And the way we've got to study those things The way we got to study those is with techniques that are non-intrusive and non-destructive. Well, that's one of the reasons why, I don't know why it has to be about me. It's one of the reasons that I started using Raman spectroscopy, because it's non-intrusive and non-destructive, and I can do chemistry on the minerals and the organic matter. It's the same way with confocal laser scanning microscopy, where I can see the morphology in 3 dimensions without disturbing the stuff in the rock. It's the same thing that we do fluorescence spectroscopy, because it's not going to mess up the thing. The problem with secondary ion mass spectroscopy is you put a little pit down on the thing, and NASA won't let anybody do that unless we find some fossils in it, in which case they have what is called an official waiver. And they did this repeatedly when systems broke down, which you guys don't know a thing about. They just kept happening and happening and happening as we were in there, presumably being exposed to pathogens from the moon. And nobody could figure out what to do about them. So we got an official waiver, and they let us go home after 6 or 8 or 10 or 12 hours in that place. I mean, it was a mess. Anyhow, I'm not going to tell you about that one. But in any case, those are the techniques. And the final thing I'll say is that for the first 3 years after those specimens will come back from the Mars, when they do get back here, probably in 2030 or thereabouts, they're going to be put into a biological containment facility that will have at least class 5, by then class 7 containment. People, you go in with glove boxes, but they essentially wear a spacesuit. The biologists will be in charge for about 3 years before they convince themselves that there are no pathogens from Mars that are going to eat up the rest of the world. It probably will be housed at Fort Detrick, because they already have such facilities, because they do bacteriological and gas warfare and stuff like that. So they have these containment facilities. Once that is done, then they will be released to the world scientific community, and that is when you're going to get some science out of it. So you can't get too anxious at this point. And the same thing happened for the lunar samples. But sooner or later, when we find there are no pathogens and all that, then we're going to learn some stuff. So I know it's a long answer, but it's a difficult problem. And it's being sorted out, incidentally, by the international scientific community, not just the United States. There are lots of people from Europe and from Russia and various other people, Japan, China, all that involved in this. And so I hope it'll be done properly.